All right, in the interest of time, why don't we go ahead and get started. Welcome everybody to WIP today. It is my pleasure to introduce Dr. Kun Huang. I asked him how he would like to be introduced. I could rattle off many titles, but instead he just wanted me to say that he's up on the third floor here at Regenstrief. And I will just add, Kun has been here for a little over 2 years, and what a 2 years it's been. So Kun, take it away.
Okay, great. Thanks a lot for coming. And since this is really work in progress, I don't want to be, super formal, but it's really just to share some thoughts and something we've been doing the past 2 years, something I learned the past 2 years, right? So when we talk about machine learning or AI nowadays, surely you hear the word deep learning all the time, but turns out for me, it's actually, even at least for myself, it's actually a learning process. By learning process, I mean, actually, I started with being very resistant to deep learning. And, but then the past couple of years, essentially, I'm trying to make peace with it in my conscience. So, I mean, deep learning surely is the background for deep learning is really underlying principle is mostly neural networks. And for artificial neural networks, right? So the word artificial comes in, that means that we are trying to develop some models mimicking the real neural networks that's going on in your brain or nervous system. So even back in I think 1940s, there are models for those artificial neural networks. But what's kind of really made it widely, have the high visibility is this perceptron model. So you think about it, we have neuron, and the neuron has other input from other neuron and different weight. I mean, I think the model behind it is in the nervous system, we have different neurons. They're like synapses connecting them. And so the strength of the synapse is kind of becomes a weight being modeled here. And then all the information comes together in, if that synapse receives enough input, I mean, you essentially, biochemically, that means you got the transmitters bind with receptor cause a channel to open, you got ion flow in, and you got enough change potential in the voltage, in the cell membrane, then suddenly you got this burst of action potential. So that's become really a 0 and 1 process. So the outcome, the model is you got those input in, and then when you have accumulated enough things based on the sum of the weights, then you got a 0 and 9 response. But to mimic it instead as a continuously differentiable function, you use a logit function instead of step function. So I think originally the model was proposed by Frank Rosenblatt. So by that time, that's essentially, this was back in 1960s, and suddenly you got this hype of, artificial intelligence. People, I mean, in every journals, every newspaper say, okay, we are going to solve it. We are going to have machines mimicking human or maybe do better. One of the example is this really optimism that, computer vision can be solved in the summer as a student intern project, right? So they have this summer vision project in MIT. Of course, then later people realize that shortcomings, but interestingly, actually part of the hype was this publishing book called Perceptron by Marvin Minsky and Parvent. But actually the book shows the perceptron has some fundamental limitations, like it cannot really do this logic operation XOR. And I think also interestingly, the Marvin Minsky and the Rosenblatt, actually they were high school. They came from the same high school, like 1 year apart. And this is maybe a lesson, just to be nice to your high school friends. So partially, I mean so this book actually helped the AI go to the first AI winter. Actually, DOD pulled out the funding from AI lab from MIT, and Rosenblatt actually died tragically in a sailing accident. But then people actually were not really stopping there, so they keep working on it, right? So, and in the 1980s, things kind of coming back. There are many different things. I mean, backpropagation algorithm were introduced, and recurrent neural networks, and later converted into, evolved into the so-called long short-term memory method. And interestingly, actually in 1987, so Marvin Minsky republished the Perceptron book. I didn't read that book, but from what people told me is really the major changes, they redrew every figure using computer. So, but instead of changing much content. But that also, if we remember 1980s, then this word artificial intelligence comes back again, and we all remember that. But then it's kind of like goes down again. In 1990s, even though a lot of research has been going on, but I think more other, theory in machine learning, especially, with the learnability and VC dimension, the introduction of those, and the support vector machine actually got a lot of attention. And support vector machine, in a way, is actually doing something very similar to perceptron is doing. It's trying to find the linear classifier of the data. But the good thing about support vector machine, I don't have the figure for that, is since it's clearly trying to find a linear classifier, and the linear classifier means the linear functions of the hyperplanes are linear combinations of features, it's very interpretable, right? You got a very intuitive to say, okay, I have my data separated here, I have a line separating them, or I have a hyperplane separating them. So the challenge is not all the data can be linearly separated. And then in order to do that, then the trick is, okay, you do some nonlinear transformation of the data, lift it into a higher dimensional space, and trying to separate your data in a higher dimensional space. So this nonlinear transformation usually is called the kernel function. So actually, once you start to do kernel SVM, then you're actually working on things nonlinearly anyway. I think a big comeback of the neural network is actually from 2006, but really have high visibility since 2012, right? So when this AlexNet won some big competition in image recognition, it's like once it got to the first, the ranked number first, it's more than 10% better than the second and other algorithms using a very complicated, many layer network. So each block here means many layers of the neural networks. So it's clearly it's very hard to train and requires millions of training samples. But nevertheless, people just got so excited about the performance of the deep learning and the potential of the performance of deep learning. So the past almost a decade, we have witnessed this really big adoption in the entire field of the machine learning, not just machine learning, pretty much many engineering field are switching to deep learning or learning-based methods. Part of the reason is, one thing is if you think about it, you got a neural network so complicated, there is no way you could compute it back like 20 years ago or 30 years ago. But now we can, especially with the implementation on GPUs. And, I have a friend right now, like I think many of us struggling to get GPUs, but you got to work with. I have a friend who used to work in NVIDIA. He was like, if you need GPUs, I have about 600 cards, but if you need more, I have a few thousand on the cloud, right? So once you have those kind of computing power, you can essentially really do whatever you want on things like this. Another is really the data. We have so much training data, especially labeled data now for certain applications. And so that really enables things, this to happen. So that's partially really driven by that. But I would say, I still think, it's kind of like, in a way, it's a little bit too hyped. Give you an example, right? So CVPR, this is a conference, it's one of the major conferences in computer vision and machine learning. It's called Computer Vision, IEEE International Conference on Computer Vision and the Pattern Recognition. Even though it's an international conference, always being held in the US. When I was a student, the CVPR, actually the entire computer vision community was really struggling. For example, I attended the conferences as a student in 2003 and 2004. This is attendance. Actually in 2002, we couldn't even organize a conference. We couldn't even find the sponsor and people to organize a conference. But you look at this year they got 5,200 submissions. Full paper submission, you're talking about those double column 10-page submissions, right? And they had, it's very competitive, so they admitted about 25% of them. And not only so, you look at the attendance. Why I attended in 2004, it's like in Wisconsin, Madison, I remember, 2004. In 2004, it was in DC. 2003 was in Wisconsin, Madison. We got about, 700 people. This year, 9,200 people attended the conference. And actually, I was told the registration site closed in like 10 minutes or something. Once it's opened, like people registered, sold out immediately. And unless you are author, otherwise there's no way you can really get a ticket to the conference. And also I remember the conference used to be really poor. We go to the banquet, like you got 2 pieces of chicken. But this year they got more than $3 million of sponsorship. So, I mean, this is, I wish I stayed in this field a little bit longer than I did, but also that to me, that's a warning sign, right? So you look at it, it's become very homogeneous topic. It's essentially the major keywords, deep learning, network, right? Working on image, neural. And so it's like, okay, you got 5,000 papers pretty much all on the same thing. And not all on the same thing, but many of them very, very, very related. To me, it's not necessarily the best thing. But also on the other hand, that means, especially if you think about the sponsorship, that means industry is really in need of such technology. But that said, I personally have resisted for a long time because, oops, I don't like it. Just because I don't think that's actually how neural system works. I mean, I have shown this before in my talk. I just want to show this again. This was why I was a neuroscience student. I don't know if it's coming or not. Okay, so when I was a neuroscience student, actually, I took the photo with my lab mate more than 20 years ago. So we were studying on this sea slug, and this sea slug is trying to eat another sea slug. It's like a monster trying to eat it. But the thing is, the purple thing actually has a sting cell and has sting cells. It stung the monster and it spit it out, right? And so I say, let me see if I can accelerate this a little bit. So just turn away. And the next time we put this back, it senses the existence of the other one immediately turned away, right? We trained this only once. We don't need to train this like, 500,000 times or 100 times or twice. So we trained this only once and it just learned. And it's based on its neural networks, right? So it doesn't really have a formal brain to have a few ganglions, but we only trained it once. And also, when you look at this, this is really a highly simplified model based on linear. Actually, the summation here was really going on inside here is very, very nonlinear, and there are a lot of local behaviors of the transmission of the electric signals. And also there are a lot of interactions between the neurons that really cannot be or haven't been captured by the current neural network models. So, but to me, really the challenge is, there are practical challenge. One thing is, generally, deep learning models needs a lot of training data. Of course, now things start to, people start trying to use less and less training data. But for us, in biomedical applications, many times, intrinsically, we cannot get those, right? So especially now, for example, I'm working with a pediatric cancer group, and pediatric cancer, by definition, rare disease. I mean, the word rare means you cannot get many training samples. And sometimes we got, the first time we worked with a pediatric group, we got about 12 terabytes of data. Turns out all the 12 terabytes data were for one patient. Right? So there were no training there. It's just one patient. So, but also I think the challenge in there is the lack of interpretability. One thing that deep learning, this should be traditional machine learning because deep learning really is part of machine learning. But the deep learning, what they taught it about is, they say you don't really need to define features. You can do this end-to-end thing, right? So you got this magic box, you threw in there a bunch of images, it will spit out to tell you what they are. That's great. But when we are doing biomedical research, that's not what we want. We want something more, tangible. For example, a big part of my research is on computational pathology, right? So we try to get the pathology images and trying to identify certain features that are related to, patient outcome or related to genomics. If we only have something end to end, we will not really learn much. I mean, to me, it's very the knowledge. Actually, I have reviewing some grants. The grants claim the neural networks are the knowledge, right? It is the knowledge. Actually, I reviewed the grant on knowledge database. Knowledge database for knowledge, or like how do we curate that? Turns out it's a database for curating different neural network structures. To me, that's a good idea, but at the same time, I mean, when I work with my collaborating pathologists or something, how do I tell them? I cannot tell them this is a box you can use, possibly will work. There's no guarantee. And so, but instead it's really what's the features you can extract, right? So for example, if we got a piece of tissue, what we want is, okay, what's the area of the nuclei? What's the shape of the nuclei? What's the distribution of the nuclear area over the entire slides? I mean, those things is much easier for us to grasp. Or we can extract the things like, okay, we have different types of cells, and in the slides, we can carve the space like this, and then define the neighborhood between type A cell, type B cell, and how often do I see the type A cell and neighbors with type A cell? How often do I see type A cell and neighbors with type B cell? And potentially they could be used to also to predict patient outcome. And once you have those information, you can go back to say, oh, it sounds like once you have the more mingled lymphocytes with epithelial cells, the patient may have, shorter time to live, but if you have more condensed pockets of lymphocytes, then the patient have longer time to live. So those are the things it's much, I think, at least at this stage, for biomedicine, we really need those, right? So for example, and this is another paper we published just in August. I mean, this is for breast cancer, and you have all those different features. Clearly, you have the cell nuclei with large areas, or you have the cell nuclei with a large aspect ratio. It means the nuclei is elongated. So you can identify those areas, and you can calculate the percentage of those areas in patients, and to see how do they are linked to patient outcome as a hazard ratio. But it turns out the patient have larger regions of cells with large nuclear areas, they do worse. They have a higher hazard ratio for breast cancer, while the patient with the smaller distance and among cells has actually doing better, because actually there is a biological interpretation for that. Is the cells with larger area usually those are the tumor cells, and you have denser tumor cells, and that's not a good sign. At the same time, the cells who are denser in this case, many times in breast cancer, that is actually the pockets of the small lymphocytes, and once you have more of the small lymphocytes, many times indicating their cancer immune system is more active and could lead to better outcome. So, and furthermore, we can actually take those interpretable features and to correlate with genes, and then we can ask questions to say what are the common things among the genes. For example, for the breast cancer, one thing is just look at the genes that correlate with higher percentage of large nuclei cells. And interestingly, actually it's very interestingly, the genes are not really cell cycle genes or genome stability genes as we expected, actually mitochondria metabolism genes. I mean, the signal is very strong. I mean, when we do, feature tests, it's like 10 to minus 40 p-value. I don't want to emphasize too much, but that's still a pretty strong signal. On the other hand, biologically, now it's becoming interesting. Well, we are talking about mitochondria. At the same time, we are talking about nuclear size. Why mitochondrial metabolism changes associated with nuclear size? I don't know. Similarly, when you look at those genes, quite a few genes actually are concentrated on a specific cytovant on chromosome 16. Why chromosome 16 are linked to this nucleosides, I still don't know. But one thing we know is chromosome 16 P13 band, exactly this band, is actually a marker for luminal A, luminal type of breast cancer. So generally, yes, I got a lot of things, don't know, but that's exactly what bioinformatics is for, right? We look at data, trying to generate something interpretable, but those become a testable hypothesis that we can focus on in the lab. Well, this is something I want, right? So I want something interpretable, I can have full control, but at the same time, there are certain things I cannot resist. I mean, people all say, okay, the neural network interpretability is horrible, but the performance is really good, right? So I mean, at a certain point, we say, okay, we cannot live just with just better performance. So for certain application, now we start to adopt deep learning. And so this is a paper just submitted last week by Zixiao sitting in there. And so we'll just using, I mean, if you want to know the details, talk with her because I really don't understand the complicated network structure, neural network structure. But the essential to say, by applying this, we can do a very good job in detecting the tumor lymphocyte in high accuracy. But the good thing is once we detect those, even though it's a black box for us to detecting those, after we detected the lymphocytes, we can still quantify it as a salient feature. And actually, we correlated the percentage of lymphocytes with the genes. Turns out some of the top genes are like CLTA3 and PD1. I mean, if you follow the trend, last year Nobel Prize on cancer immunotherapy was exactly given to those two genes, right? So that really shows, yes, we can still use it, but instead of do the end-to-end thing, we try to make it a component of our research, then leverage the accuracy. So this is another work by Zixiao, and also using the neural networks and trying to separate the stroma and epithelial regions in breast cancer. And the left side, clearly you can see those are the original images. The middle side is actually human, the published human annotation. And this is an example, it's a region actually missed by the human, but easily detected by the algorithm. So potentially showing the algorithm really works. But besides its performance, sometimes actually turns out we can also use it for exploration. So we have another student and in the group, and he's very interested in deep learning. So he worked at Jeremy, and so he worked on trying to develop a neural network model for integrating different data to predict patient outcome. My biggest concern is, can you give me something out of it, right? You can have a model to say, okay, here's patient information. I predict patients in high risk or low risk groups, good. But really, what are the features? So finally, he did a compromise. We're like, okay, I think that's, you can talk with Jeremy Moore sitting in there about it. And the idea is, okay, after you train the network as hard as you can, you start to take out the features one by one, and to see how bad it will be, and using that, how bad it became as a way to rank the features. And so clearly you can see there is one feature after you take it out, it's just the performance drops dramatically. And when we first met, and the question is what this feature is, and the feature is age. So it's the age of the patient. Turns out that among the entire model, the age of the patient has the biggest influence on the outcome. You know, it's right, because we know for cancers, age is the biggest risk factor. But then the question is, what can we do with it, right? So because then if like there are two patients, both are 60 years old, can you do anything better? So one question would be, can we stratify the patient based on the age? So instead, using the model, then if we stratify the patient into three groups, like 50 and under, and 50 to 70, 70 and above, and can we do anything better? Turns out, it's pretty interesting. So for the younger group, there is, a factor. Turns out to be progesterone receptor status. I mean, progesterone receptors is one of the major markers, for breast cancer, especially for triple negative breast cancer, when you have ER negative, PR negative, progesterone receptor negative, and HER2 negative. And it happens a lot more in the younger group. And for the mid-age group, 50 to 70, the single most important feature is ER, estrogen receptor. Right? This is the time when menopause starts to happen and estrogen level changes. But then for the older group, it's not PR, not ER. It's actually different features related to molecular, different molecular profiles and different molecular signatures, genetic mutations. So even though, yes, maybe we have a model that is not a perfect model, but I think what this really shows us is when we are doing, biomarker studies, we should maybe stratify this to patients' age-specific biomarkers instead of just, okay, I have this general markers, right? Another study what also Jeremy did was also interesting, and he was trying to develop an algorithm to predict patient outcome and compare with two other algorithms in the field recently published. And he applied this to 14 different cancers. And when you look at the performance, frankly, there are not too much differences among the 3 different methods. Sometimes, one method do better, sometimes the other one do better. But what's really interesting is the performance is actually very consistent among the methods, but they change a lot among cancers. So one question is, so what's the difference in cancer? This is based on gene expression data. So one thought is, okay, this is suggested by another student, Travis, and he was like, well, then how about if we also look at the patient total mutation burden? So we correlated with the average total mutation burden of the patient. Actually, generally, it's a negative correlation. If you think about it, it's actually a very direct message to say total mutation burden is DNA information, right, from genotype. Right now, the model is based only on RNA. We are not using the right data for certain cancers. And for cancer like kidney cancer, I mean, it has a very low total mutation burden, and generally RNA-based model work well. But for some of the cancers, when they have large mutation burden, regardless of how good your algorithm is, it will not work because you are not using the right data. It's just very straightforward. So that actually teaches me something. Okay, so deep learning, actually, we can use it to do exploration. It's not just a black box. I mean, surely its own component is a black box, but it's not just a black box alone. And it turns out, I think another way to think about it is it's also convenient. Because if you really think about what deep learning is trying to do, you got this neural network model. Essentially, you say, I have a bunch of data coming in, I try to fit it with the label I know. So it's really trying to optimize the error. So really what this entire black box does is trying to do a nonlinear optimization, right? Pretty much any nonlinear optimization tool is a black box. So if I think that way, I can live with that. So, and this especially if you sometimes have competing or multiple objective functions and you try to do them together. And especially for this case, when we have the different, on the left is not from us, I'm just to show the single cell data. And when we have the single cell data, one thing is you got different subtypes. And the challenge is when you have different subtypes you want to match, that becomes very challenging, right? So, and in this case, actually, even more challenging, I think, is we're trying to match the human and mouse data, brain data. So some cell types can be matched, some cannot. Or sometimes for human, you have much refiner subtypes, but for mouse, you have a general clone. So now you try to balance different things and trying to essentially do a gigantic optimization function. And now, if you want to use a traditional way, you can. It's just going to take a lot of math and computing to do that, a lot of math to do that. And not necessarily always you can do it fast. But using neural networks, that becomes just convenient. It may not guarantee a good solution, but many times it magically just do reasonably well. And this, I mean, this is another example by Tongxin, and a similar problem when we have single cell from multiple samples, and how do you map them with different subtypes of cells, and using actually a learning scheme, essentially you try to say, I want to represent the data here in this layer, but make sure during the middle when you have the low dimensional representation for the 2 data types, they are consistent. And so you have the cost function to say partially is on the top and on the bottom, trying to make them consistent. But in the middle, you try to also make the transfer between the 2 data sets are consistent too. So you got like 3 objective functions you try to balance. Instead, you say, okay, now I'm just dumping everything in there. And then turns out it works remarkably well. So just last minute to summarize, this is what I learned the past 2 years. We always have to do the trade-off between accuracy and interpretability in biomedical world. So depending on really what you want and what's your situation, we can choose to use deep learning or not. Or maybe to make a deep learning, consider deep learning as a nonlinear optimization and make it a part of the workflow. Like we have another paper, it's really on using canonical correlation analysis, but we use deep learning as one component to do the clustering in the between. Sometimes I'm still learning generative models. People have been doing this a long time. I never paid attention. I'm like, this has nothing to do with me until last month I saw a paper in Nature Methods. They're using generative models to study single cells. I'm like, that sounds interesting. So I'm into that. But I think still we need to periodically go back to the basic mathematical principle behind it. 2 days ago, my PhD advisor sent me a paper from their newest paper. So what he has shown before actually is you can do those multi-layer neural networks, essentially do the nonlinear transformation. But instead, if you do that kind of like magic transformation, if you just do PCA every layer, actually you can achieve very similar performance. But then you got much more interpretability. So just want to thank people who really contributed work, and thank you very much.